Thank you, everybody. Thank you very much, Dr. Bhatt and Dr. Ashraf, for that kind introduction. So we're going to get started. I think we have a good crowd today. 43 of us have already logged in, despite being a Saturday evening, and we will be recording, and all the recordings will be also available through YouTube, Facebook, and other social media channels. And as we know, as we speak now, world has seen 94 million people being vaccinated, and Israel has vaccinated 35% of its population, and we are eagerly waiting for data coming out of Israel to know the efficacy. And we are also bombarded with terms like vaccine nationalism and tribalism. So we're going to hear more about all these concepts and advanced questions regarding our vaccination campaign. And many of us are involved in educating public and also our patients on an individual basis. So it's very important that we have our knowledge updated with reliable sources. That really is the aim for today's webinar. With this introduction, I'm going to invite Dr. B. Madhavan Pillai. He's the past president of AKMG, and is specialized in internal medicine, hematology, and medical oncology, triple board certified by the American Board of Internal Medicine. He's a clinical professor of oncology at the Sidney Kimmel Medical College of Thomas Jefferson University, and he's also the president and CEO of International Network for Cancer Treatment and Research. And Dr. M.V. Pillai was also a leading member of the Kerala's COVID-19 task force. And he's also a senior advisor to the Global Virus Network, and he's a graduate of Trivandrum Medical College, something we all are very proud of. And Dr. M.V. Pillai is going to introduce our first speaker for tonight, Dr. Shyam Kottilil. Over to you, Dr. Pillai. Thank you.
Thanks, Nisha and Subra. And I think I can identify Maji, Aravind, and maybe Sindhu, and Nikhil also is here. Okay, so a lot of people are personally known to me. My job is very simple. I won't take too much time introducing the main speaker in terms of the association with virology today, Dr. Shyam Kottilil, MD, PhD. Now, Shyam and myself, we have been working together for the past three and a half years to establish the Institute of Advanced Virology in Kerala, which is connected to about 52 such centers around the world, distributed in about 32 countries. An idea which was proposed by two internationally renowned virologists in 2012 as a warning that the world is going to be threatened not by nuclear war or earthquake, but by recurring epidemics from viral infections, which we haven't identified yet. And their predictions came true. Dr. Robert Gallo, with whom Shyam is working, and is somebody okay, and okay, and also Christian Bershaw, who is currently the president of the International Global Virus Network. And they proposed this, and the Global Virus Network came into existence, and we joined them 2 years ago. And we are happy that we are getting affiliated to the center in Japan and also to the center in Ireland. Now, let me read out the profile for Shyam. He is now co-director of the Institute of Human Virology Clinical Research Unit. And he's also the division head of infectious disease, University of Maryland School of Medicine. He served as the director of the Clinical Trials Unit, Division of Infectious Diseases at the Institute of Human Virology. And Shyam received his MD from Trichy Medical College, PhD in Immunology of HIV from Memorial University, Canada. He completed residency from Brown University and did a fellowship in infectious disease at NIH with none other than the celebrated Dr. Anthony Fauci. So he is the emissary of Tony Fauci with the AKMG group. Now, Dr. Kottilil has conducted several pivotal investigator-initiated trials in hepatitis C, and he has authored over 300 peer-reviewed publications. He has received numerous awards, including National NIAID, that's the National Institute of Allergy and Infectious Disease Meritorious Service Award for Innovation in Science in 2014, and another NIH Outstanding Mentor Award in 2013. Dr. Kottil has mentored over 30 MDs and PhDs during his academic career. Incidentally, William Hall, who heads the Virology Institute in Ireland, Shyam and Christian Brasheau, who is the current president of the Institute of Virology, and not the Institute of Virology, Global Virus Network, are heading our scientific wing of the Institute of Advanced Virology in Kerala, which is just taking shape. Fortunately, we got a nice guy by name Abhijit Banerjee, who was formerly the director of the National Institute of Immunology at the JNU University in New Delhi. He has taken over. He is recruiting staff, and the laboratory is being set up. Hopefully, in the Kerala pace of getting things done, it may take 10, 15 years, but it might definitely come up sooner or later. I want to congratulate Subra, because what fascinated me in the newsletter is the picture of the Matryoshka dolls from Russia. Some of you might remember that AKMG took a tour of Russia a few years ago, and I was fortunate to accompany them. And the Matryoshka dolls symbolize three things: family, legacy, and fertility. And all these three things are highly commendably reproduced in AKMG of 2021. The family is intact, it's actually growing. The legacy is outstanding. The fertility, as assessed by the increasing number of life members, is also very encouraging. So I don't know, whoever chose, maybe it is Nisha or Nigel, who chose the Matryoshka doll, did the right thing to do. Now, two articles which I read today may be of importance in today's discussion. One came in New York Times about a study from UPenn about pregnant women transferring the passive immunity to the newborn. So pregnant women who got the COVID vaccination gave birth to children who have antibodies confirmed, and neither the child nor the mother had any problem. A study came in JAMA Pediatrics and quoted it in all leading newspapers. The second study is probably of poetic significance to all of us. It comes in The Economist, how France is struggling with COVID vaccination. A popular survey of the French people revealed that only 42% of the people are interested in getting vaccinated. As we all know, there is a strong anti-vaccination group in Europe, and France is no exception, which takes us back to Kerala, our home state. In 1813, when Ed Jenner was introducing vaccination in England, the church and the society and even the medical establishment were opposing his move. The vaccination was not received with warmth or enthusiasm. Right at that time, two nations accepted the smallpox vaccination with a lot of enthusiasm. And one was Kerala, the then Travancore province. The queen at that time is Gauri Lakshmi Bai, 1813, more than 200 years ago. She came out from the palace to raise all the misconceptions about smallpox vaccination and publicly vaccinated herself along with inmates of the palace. And remember, this is at a time when England was up in the arms against the vaccination process. Later on, we know that Louis Pasteur, of the pasteurization fame, and also who discovered or invented the immunization against anthrax and rabies, was not received with a lot of respect initially. The French people were against vaccination, and they continue that even today. That's why Economist is reporting 42% of the French people are only happy to take this vaccination. And compare this to Kerala, where people are fighting with each other to get the vaccination as quickly as possible. So in 200 years, it looks like the French have not changed much in their attitude towards vaccination. There are a variety of reasons, and I don't want to elaborate on that. So this is the contrast of how the Europeans are handling vaccination. At a time, Travancore and later on the Kerala has come out successful, many of the vaccination programs, and quite enthusiastic about it. So hopefully, with the emergence of the Institute of Advanced Virology, which is the only center in India connected to this 52 global virus network, we may be miles ahead of the rest of the world in the years to come. And I'm so happy and so proud to introduce my colleague, Shyam Kottilil, as the next speaker of today's symposium. Shyam.
Thank you so much, Dr. Pillai. Ever so gracious and so elegantly dressed. And I have to say that I have forgotten how to wear a tie anymore since the last year because we have not been doing. So thank you for the opportunity for AKMG for giving this short presentation. The idea is to highlight some of the topics that have been controversial or confusing. Not that I have all the answers, but at least just to talk to you about what the data looks like and what the data is supporting at this time. For the CME purposes, these are my disclosures. So the objective is to understand viral evolution process that happens today and the impact of the new mutants on the current pandemic situation, as well as current and future vaccination strategies. We'll also touch base at the end, talking about a little bit of a non-immune pediatric population and its influence on how the pandemic is going to be resolved over time. And I just want to start out by saying that highlighting some few important points. So the studies of the two vaccines in the United States with the Pfizer and Moderna have shown to prevent symptomatic COVID infection. And that's very important to understand because they did not test for people with the asymptomatic infection. But so you really cannot say that it will prevent all infection. But it's cautiously, one has to understand that we probably will get infected with a mild disease. It does have a disease modulation effect. That means that people who get the vaccine and then eventually get a coronavirus infection will have a milder disease. So there is a disease modulation effect. The fact that they did not test people every week for nasopharyngeal virus and PCR them for coronavirus, or all the 40,000 people, we really cannot tell it could prevent the infections in these patients. So that is something to understand, that you may still get asymptomatic infection and could transmit to other people. Think about that and be able to use that social distancing continue even after getting vaccination. So vaccination, the question is that should we check antibody titers after vaccination? There is not recommended to check antibodies because we don't know what to check. There is no correlate of immunity of this vaccination so far established. We hope that subsequently in the next few months, we may be able to understand and correlate something like an antibody titer against spike protein as a correlate of the protection, but we don't have that, like we have for hepatitis B or influenza. The long-term efficacy is another question that's been quite debated in social media. How long is the efficacy? We can only establish efficacy as long as the duration of follow-up. Studies are very quickly done and the people have been followed up. So over time, we may be able to follow the people who are participating in the studies and see whether they are going to get infected or not. However, the important factor is that there won't be any placebo group, right? You really have to look at infection in the vaccinated group and compare them to the placebo group to say how much of a protection is given by the vaccination, because the placebo group will be vaccinated. So most of those people will not have a good control group. So then it is going to be a little bit of unclear how definitively we can establish what the long-term efficacy of the data is. You can, however, look at the infection rate in the people who are vaccinated. So we'll get some idea about that. So if you are infected already with COVID, what is a strategy to get vaccination? The general recommendation is based on the best clinical judgment. That's because we assume that if you're infected, you're going to have natural antibodies. And this presence of these natural antibodies at high titers will interfere with the vaccine uptake, and the vaccine's efficacy will not be there. Natural infection may not protect you a long time. We don't really know. So the best clinical judgment is I wait for 90 days from what we know of immunology of SARS coronavirus at this time. we know that it goes down, and that would really lead to the vaccine uptake. And that's not based on evidence. We don't really have a lot of evidence to support this, but that's the best judgment from the CDC. So next concern that people have is that what about the delayed dosing schedule that has been propagated in the UK and some degree during the past administration, saying that we are running out of the. It is not a scientific decision. We got to follow the evidence. The evidence was done that Pfizer vaccine, the second shot was 3 weeks after, Moderna vaccine 4 weeks after the first dose, and that's what the data is. So a scientific decision is not delay second vaccination. It's a supply demand issue. It's a political issue that people may all have vaccine. They're going to have to prioritize how to use the existing stock of vaccine, but that's really not based on scientific evidence. Scientifically, we cannot recommend delaying the second dose because that's not what the data dictates. So the efficacy of different subset of population, most of the studies have done and powered for general population, not for subset, not for ethnicities. And if you break down the data on different subgroups of people, the confidence intervals are going to be so large, and you really cannot make any clear distinction of whether efficacy on different subgroups. For all practical purposes, we had to assume that the vaccine works very well on everyone because it worked everyone in preventing symptomatic infection. And until we do subset studies on different countries and different ethnicities, we will not know for sure whether the efficacy is similar in all ethnicities. So if you have a genome, we will mutate. That's for sure. And there are studies who clearly establish that the bigger the genome is, the mutations, the rate of mutation is lower. So if you look at human beings, we are here. We have a big genome, and fortunately, we mutate very less. So over time, the smaller the bacteria are, the bacteria mutates more, double-stranded DNA like hepatitis B mutates quite a bit more, and RNA viruses that we talk about SARS coronavirus, which is the beta coronavirus, also mutates quite a bit. It doesn't mutate a lot. Not all RNA viruses mutate at the same rate. Like HIV and hepatitis C probably mutate a lot more than coronavirus, but it does mutate because of the amount of virions that be generated, the capacity to evolve against the pressure is higher, and because of smaller mutational genome. So what do we know about mutational protein? I want to take one minute on explaining the 3 different types of mutations that we have. You have the wild type variant. That means it's exactly as it was supposed to initially described. And over time, mutations can occur at different parts of the genome. And some of these genomes may have no impact on vaccines or treatments. We call them sensitive mutants that nothing to do with the replication capacity. They're just accumulated in tandem. Then you can have mutations in different regions that confer certain characteristics. That means it may bind to the receptor better or become resistant to treatment, or it can also escape vaccine. And those are the resistant mutations. But one has to remember that when you have mutation that is changing the virion, it also affects the natural attributes of the virus. Sometimes it may become less replication competent, which we call it the fitness of the virus is reduced. And if such fitness of the virus is reduced, it doesn't really have a selective advantage to spread very rapidly. What we worry about is that this group of viruses, as they evolve to people, in fact, from people to people, from country after country, they are going to evolve and finally adapt against the pressure that they can now have resistant viruses, probably against the vaccine, and also have characteristics that increase replicative fitness by binding to the receptor and replicating well in different people. And that is something that people worry about. That means you by chance, because it's going through billions and billions of copy numbers, you could get evolved into that group. Because these mutations do occur. That's not something that we're very concerned. But the fit resistant mutation is something that we're concerned about. So what happens with SARS coronavirus? Does it mutate? Yes, it does. It's a good news and bad news. The good news, this is SARS coronavirus is about 30,000 nucleotides. And each position is a nucleotide. Good news is that there are only the very small regions that are very hypervariable. This is the data of the variations at different parts of the genome that has been reported so far. So you have region here, region here, region here. Many of these regions have multiple mutations, a more preponderance of mutations accumulate there. Of critical benefit is this region, which is the spike codes for the spike protein. As we all know, the spike protein is the main protein that's been targeted for its preventive strategy for vaccine, as well as monoclonal antibodies for prevention and early treatment of mild to moderate diseases. So this is of critical advantage. So this is a mutation, D614G mutation was presented very early in the epidemic, and as an example has been followed over time to see how rapidly these mutations can evolve in this particular region of concern. So this is what it says. The mutation D614G was first described in Scotland in January of 2020. By March, it was present in 60% of all isolates. And by June, the summertime, it was present in almost all isolates. So this is exactly what happens in terms of a pandemic with an RNA virus that has the capacity to mutate quite a bit. So it has an adaptive characteristic that makes it easier to spread and adapt and then move from people to people across the country very, very quickly. The rapidity that it requires is completely amazing. And this is something that people are concerned about. So the other concern is, which we have, is that when it mutates, what will happen to the? We also know that it really needs the spike protein to bind to the receptor. This is a three-dimensional diagram of the virus when it binding to the receptor is angiotensin converting enzyme 2. So this is the receptor binding domain that binds to interact with the domain of ACE2. It's a complicated figure. What I'm telling you is that if you mutate too much, that will affect the characteristic of the virus to bind to the spike protein. So most of these random mutations will not give selective advantage for the virion. So they actually are not going to spread. Unfortunately, or once in a while, you're going to get a variant like the South African variant that we have, has great affinity to bind to ACE2, and they can actually replicate better. That is the natural selection. So the question is that how does that variant impact our current vaccination strategies? So these are concerned because we just take time for us to understand whether vaccines are going to be effective in this against all the different variants that we've described. We have to understand that when people say that, hey, this vaccine works against variant, what they really mean is that it works in the laboratory. What do they do in laboratory? That you can actually take cell lines or hamsters in vivo and then infect them with SARS coronavirus. The cell line usually uses the monkey kidney cell line or virus cell line that can be infected with SARS-CoV-2, and they can replicate there with the wild type or the variant. You can then do a neutralization assay. That means you can neutralize the virions produced by these cell lines by using a control serum or the serum from somebody who has been vaccinated. If you take that vaccinated serum for the Pfizer vaccine or Moderna vaccine or the J&J vaccine and then you're able to neutralize it very quickly, you can see that do you make enough antibodies, and these antibodies induced by the vaccine can prevent the cells from getting infected. And that's what most people do, and they'll say our vaccine works. This does not really mean that the vaccine protects you from getting infected from all the different variants that tested here. And that's a big leap of step. This is an important first step in our understanding of how variants react. If you've read newspaper today, there were two really important data that came up. One is, again, the public release. No one has seen the data. There are two new vaccines have been recommended in the UK, Novavax and J&J. They are really found to be very efficacious. They actually add to the armament of the now 5 defective vaccines against coronavirus. The Novavax was effective 90% overall, but its efficacy in South Africa was 49%. Same thing with J&J vaccine, efficacy overall was close to 70%. Its efficacy in South Africa, 57%. What we do not know is that is the lower efficacy in South Africa you've seen, is it because of the new South African variants? Only time will tell. We don't really know. But that is a little bit concerning that the vaccine efficacy in vivo may not be as good for some of the variants that come in. This is something that we have to watch for, and we have to see what impact it will have moving forward. The last point I was going to make is the pediatric population. Unfortunately, in the United States, we are guided by the Title V of the FDA. That requires they have two different things. One is a PRIA Act, which many of you are familiar, that if you have a new medication, as a new investigational agent, you have to establish a separate safety and efficacy assessment in pediatric population. So that's where we are with the vaccine. We don't have an approved vaccine yet. So you have to do those studies. So Pfizer, the pediatric population divided into two for the purpose of coronavirus. Kids who are adolescents who are from 12 to 17, and the kids who are less than 12. They are very different because the 12 to 17 is much prone to get infected and transmit the disease to others, while less than 12 have much significantly reduced incidence of infection. So the Pfizer has already completed enrollment of a study of 12 to 17. They're moving ahead with the kids. So these studies have to be completed for it to be, this vaccine to be available for pediatric population. Hopefully this will be available in the next few months, probably by the summertime, we'll be able to immunize all people. The best pharmaceutical for children to act is for approved medication to expand indications from adults to kids. So when the vaccine is approved, it also allows you some much better pathways to immunize kids. And that would be a better strategy for us to look forward. And where does a pandemic go from here, right? And that's a question that everybody asks and everybody has an answer. And many, many times I've been wrong the last year. So we're only very limited in our expertise, but that doesn't prevent us from speculating. So what is that? Basically, when the epidemics first started, every one of us was susceptible to the SARS coronavirus because we're a completely non-immune population. The susceptible population will go down because people get disease. That's a Denmark model. You develop herd immunity, and then you just don't probably not protected against subsequent infection, or you can be protected by vaccination. The predominant strategy we're using here is vaccination. So if you vaccinate people, then you can build up that immunity over time. If you can immunize significant amount of people in 1 year globally, you can bring the virus down and achieve a control. So that is a goal that the optimistic goal is that within a year, we will have SARS coronavirus move from a pandemic to an endemic disease. It may not completely go away, but it will be an endemic disease. What stands against us in achieving this goal are two important factors. One is immunization. Dr. Pillai already mentioned that reluctance of people to get immunized. This is the big factor. If people are not going to get immunized, even in developed countries, it's going to be extremely difficult for us to achieve that goal. The second part is the variants emergence. If the variants are escape mutants, then that becomes much more critical for us to develop vaccination strategies. We may have to think about booster strategies. We may have to think about new variant vaccination strategies to do those studies quickly to make sure these variants are under control, and hopefully those variants don't come up. Those are the two strategies that we are concerned about the pandemic being sticking around for a longer period of time. So in summary, the new variants are an important concern from an epidemiological point of view. There are containment of the pandemic will become definitely curtailed if these variants persist. Early laboratory studies said that current mRNA vaccines are able to neutralize the virus in vitro. That is good news, but we just need to show that that's actually true in patients in vivo, and that would be the critical step. Further studies are ongoing. There are studies using these vaccines, so we'll be able to tell over the course of the next few months. And it's same thing with the pediatric population, because we need to, from an epidemiological point of view, we need to immunize pediatric population as a reservoir. So it doesn't really have a major reservoir to drive the epidemic while we are continuing to vaccinate people. Unimmunized people and emergence of new variants are the major challenges for containing the pandemic. If we can able to immunize people and we have been able to tackle the new variants, we should be able to contain the pandemic in about a year when most people are immunized. Thank you.